Hi, I'm Monte McGregor, and I'll be talking about the life history of the Kentucky Creekshell Villosa ortmanni. I'd like to acknowledge some of our staff and our co-authors, our funding sources, the U.S. Fish and Wildlife Service, state wildlife grants, and some of our partners, such as some of the universities and private organizations and government organizations as well, especially our families who support us during our endeavor. Kentucky is home to over 100 species of freshwater mussels, 41 genera. 27 are federally listed as endangered or threatened, 8 of which are extirpated, and we're going to be working in the Green River system with this species, which it's an endemic. Green River is located in southwestern Kentucky. It has over 100 miles of free-flowing water. 29 species of mussels and fish are imperiled in the system. It's one of the largest tributaries to the Ohio River. The diversity of mussels in the green is very good, over 150 species of fish. One endemic, Leonio ortmanni, which is the one we're talking about. We have 17 federal listed species as well, and 3 of these are extirpated from the green. The Green River has several major tributaries which harbor the ortmanni, and we'll be talking about some of these locations. So basically our study is to examine the distribution through our personal collections and also our database, and describe the habitat, observe the gravidity period, examine the adults for anatomy, brooding chambers, lures, papillae, so on, conduct host fish trials as well, and then culture juveniles. Villosa ortmanni, Kentucky Creekshell, was described by Walker in 1925 in honor of Ortman. The type locality is Green River at Mammoth Cave, and it's changed a couple of times in the names over the years. It is closely related to a couple other species, Villosa ortmanni, Villosa venoximensis, and Villosa leonosa, are very similar species, although they're present in different stages. The 3 species that are similar, you can see at the bottom, do look a lot alike, although there are quite differences in some of their papillae, their sizes, their mantle colors, their lure papillae, and even some of their fish hosts are not the same. In 2018, Tom Waters revised the genus Villosa and changed the name of Villosa ortmanni to now Leonio in honor of Isaac Lee and Arnold Ortman, and then the Unio, of course, one of the first mussels described. The ortmanni is located in mostly the upper part of the Green River in the tributaries, typically some in the upper part of the main stem as well. We collected females from 3 or 4 different tributaries, the Rough, the Nolan, and Russell Creek, and Brush Creek, from 2004 to 2020. The habitat of Villosa ortmanni is typically shallow moving runs with sand and mixed substrate with moderate flow, and you can see I've indicated 3 of them that are in the substrate. The sand is an important part, and it's typically about knee deep or less where I typically find them. Here's more habitat in the main stem green, which is a little bit larger, 40-50 meters wide, and it's mostly sandy habitat where I found. This is one of the largest specimens that I found, 72 millimeters, and it was about 17 years old. Some of the habitat associates that we wanted to test was the rainbow darter, because we typically found that species everywhere we saw the Villosa ortmanni, and so we knew it might be a potential host, so we wanted to test it. Just some of the animals we collected. We collected several that were anywhere from 45 to 72 millimeters from different river systems, and you can see sometimes they had larvae in them, sometimes they were what we call full, some were partial. There were a mixture of purple and white nacres, even some cream colored ones as well. And fecundity typically was anywhere from about 1,300 to 63,000 per female. Leonio ortmanni is a small mussel. It has these V-shaped beak sculpturing when it's young. You can't hardly see it on the adults. The males are blunt, come to a point, and the females are rounded on the ventral posterior 1/3. And typically the shells has fine green rays on it, which is typically absent in young ones. Sometimes you can see it, like these here that are a few months old, you might be able to start seeing the faint rays. The brooding chambers are typically like other Villosa, like this one's the Villosa iris. It uses the posterior 1/3 typically, although there's a little bit of gill structure on each end of the marsupial pouches. Here is the beak sculpturing with this double loop or this V-shape beak sculpturing. The males and females are sexually dimorphic. Here's the male on the bottom with the female on the top. You can see this distended area on the posterior 1/3. And here's what the young look like from the wild. Here's an example of what we see in like one of the sites in Rough River, for instance. We see anywhere from 3 to 9, 12-year-old animals. This is an example of some young ones that haven't reached reproductive age at this point. But the females, you can see this 9-year-old is definitely a female and started reproducing somewhere around 5 or 6 years old. The Glaucidia is typically subspatulate, and it's definitely higher than long. This is the length of the Glaucidia. It's about 2.5 times the hinge length. The length is less than the height, about 1 to 1.5 roughly. The standard size for these were about 311 units in length, and height, I mean, with 226 in length and about 120 for the hinge length. The Glaucidia is a little bit normal size to almost all the other Villosa, but it does have these sensory hairs, which we notice there's about 4 on each valve. Sometimes they're also in this vitellin membrane, but when they come out of the adult. And here's a small unfertilized egg, and then these blue dots are the location of these sensory hairs. These sensory hairs are chemosensory. They mostly respond to different chemicals. Here's a typical egg you see of Villosa ortmanni, about 350 microns in length. It's more of a teardrop shape or oval shape, and this was an unfertilized egg versus a mature larvae here. And here's again the sensory locations, the sensory hair. They release these placenta that are about 4 to 14 millimeters long, and you can see this has a mixture of eggs and larvae within the placenta, and this is what's released out that the fish would eat and get infested. Juveniles start off about 225 or so microns, and then before long they get up to about a month old, maybe 450, and then a couple months their gills are fully developed with their siphon anatomy present. And you can see the stomach area, the gills, and then the intestine with the papillae and the siphon already forming at less than 5 millimeters, which is just a few months old. This is more of the color they get when they're older, more of a darker orangish color versus the yellow of the younger stage. The juveniles do have these fine rays that are starting just at 5 to 6 months, and you can see the development of these animals. These are over time. With the full intestine and stomach, and then the gill structure with the papillae on the siphons. Again, more of the beak sculpturing, these V-shaped loops that are present on these young animals, pretty prominent on some of these animals, much harder to see on adults. The anatomy of the Kentucky Creekshell, basically they have about 14 to 18 distinct papillae from the branchial open. This is basically the lure of the females. The color of the mantle tends to be black or yellowish color, speckled pattern, and the marsupial pouches, there's typically about 8 to 20. Here's what the marsupial pouches look like inside the animal, and you can see that there's quite a few of these that are charged. The ovisacs are charged with quite a bit of larvae. The gravidity period we found is anywhere from 1st of September to about middle of June, and we consider these Brady tick long-term winter brooder, and they typically spawn in August, which we haven't found any gravid in August. It's a close-up of the marsupial pouches. The females have this brachial mantle flap that has papillae on it that they undulate to attract the host, and the flap on each mantle starts at the brachial opening and typically has about 15 to 25 papillae, which is more than what the Leonosa and Vinoxamensis has. So we think this is a good species. Here's an example of the papillae, what they look like with this pattern, and these are the papillae and of the branchial papillae as well, and then the papillae on the mantle flaps of the female. And here's an example of what these look like when they're moving, and you can see that undulates to try to attract the host fish. Here's another shot of a couple more females, and it undulates these mantle papillae in the flap to attract the host fish. In this case, we'll find out what that is in just a few minutes. Just a cryptic lure to attract the fish nearby the mussel. The males, the siphons in the males and females look very similar on the brachial openings. Here's the in-current siphon versus the ex-current. Definitely more developed papillae on the in-current versus the ex-current. Again, very similar from another site. You can see the dark mantle edge around the papillae with the lighter around the brachial openings. We tested 21 species of fish for the host We run trials from 2004 all the way to 2020, and we tested sculpin, sunfishes, darters, and minnows by pipetting 100 to 300 larvae on the gills and monitoring for glaucodal metamorphosis. Here's some of the species we found near the site where the mussels are found: fantails, banded sculpins, rainbows, greensides, johnnies, and banded darters. So we wanted to test these plus additional species as well. The sculpin were the main host, and you can see from these numbers. We tested 12 sculpin here, and we got 31 juveniles, 15 here, 715 juveniles, 12 sculpin here, and got 1,400. And again, another from another site, we got 365. So sculpin is definitely a good host, while these other ones are not working at all. The banded sculpin is the one that produced the most juveniles, so we think this is the natural host. It's found right where the mussels are living, and it works good. Here's what they look like on the gills, and here's what the juveniles look like when they come off after just a few months old. Again, the Lepomis megachiris, we don't think, if it is a host, it's a marginal host. It's the only other species that even attached to the gills. Kentucky Creekshell overlaps very well with the distribution of the sculpin in blue, the Kentucky Creekshell in orange, and so they overlap very well with each other, and it makes a perfect host. So in summary, Leononia ortmanni is a long-term brooder, broods from September to June. Larvae are anywhere from 226 to 311 on the length and the height. Eggs are teardrop shaped. Larvae are released in placenta. The larvae have sensory hairs, 4 per valve. The mantle edge has papillae as a cryptic lure. Fecundities anywhere from 1,300 to 60,000. Age up to about 17 years. Maximum size 72 millimeters. The sculpin is the main host, and we stocked several juveniles from where we reared these animals from cultures. We also transform these in vitro without a host, so that does work as well. And the juveniles can grow about 15 millimeters in just a few months. It is on the petition list for listing. It's imperiled in Kentucky, and it's a species of greatest conservation need. And again, the sculpin is the only known host. And here's what the juveniles look like when they're 4 to 6 millimeters in length.